Okay, let's go. So today we're going to talk about eukaryotes versus prokaryotes. Obviously, if you do biotechnology, some people might end up working on like pigs or cows, so like big animals, but then you might also end up working on bacteria that associate with those animals. So you need to have a good understanding of prokaryotes versus eukaryotes, what the big differences are. And then in terms of just molecular biology, we often work with bacteria, E. coli, versus yeast is a eukaryotic system. And you'll see all kinds of applications of yeast and E. coli in biotechnology. And one's a eukaryote and one's a prokaryote. So we want to understand the differences. Yep. Oh my God. Thank you. I would have gotten the whole lecture without anybody letting me know. Okay. And we got the PowerPoint to record, not as a crutch. Okay. Okay, so what's the big difference? That was a question on the intro quiz. What's the big difference between eukaryotes and prokaryotes?
Nucleus.
Yes. Most people got it wrong. And also organelles. Somebody put organelles, which I think that would be correct as well. But the big thing is like the nucleus. Eukaryotes are going to have a nucleus in the cell. Prokaryotes will not have a nucleus. They have like a nucleoid, which is like a region where their DNA sits, but there's no nucleus. So often I've read eukaryotes are quote unquote cells with things in them. Prokaryotes are just like a cell. So that's a major thing. If anything, remember that. And then there's also like a complex versus simple dichotomy. So eukaryotes are complex. They're more complex. There's more compartments. There's more sorting. Prokaryotes, in a way, are more simple. Although that's kind of like an aesthetic word that might not necessarily mean something in a scientific context, simple versus complex. But in general, we mean eukaryotes are segregated into tiny compartments. Prokaryotes are just kind of very, very simple structurally. Again, though, that's debatable. Cytoskeleton. Eukaryotes will have like cytoskeleton, which will be made up of like microtubules or actin, filaments, things like that hold the structure of the cell. And prokaryotes do not have cytoskeletal elements. They kind of just have a cell wall, which holds the structure. Okay, so that's one other big difference. Okay, now let's talk about, since you're going to be doing lots of genetic manipulation in the future of eukaryotes and prokaryotes, we need to know in detail the details of their differences in nucleic acid organization and structure of eukaryotes versus prokaryotes. Okay, so the first thing, size of genomes. Okay, which one of these do you think is going to have a tiny genome? Prokaryotes. Prokaryotes. Bacteria are going to have tiny, small genomes. Okay? Eukaryotes are going to have much bigger genomes. Okay? That's just a general theme that you see. So let's talk more in detail. So why? Why do eukaryotes have bigger genomes? What? Yes, they're more complex. That's logically why they need a bigger genome. Oftentimes they form multicellular organisms. Those multicellular organisms have different cells. Each of those different cells behaves in a different way, so all that needs to be programmed into the genomes. Also, they have bigger genomes because they have much more non-coding DNA. Okay, and we're actually going to get down to the root of really, really why bacterial genomes are so small. The fundamental forces that cause that. But also just a thing to note is that the bigger genomes have a bunch of non-coding DNA. What is non-coding DNA?
Like the introns?
Yes, so if you have a gene, the eukaryotic organization of a gene would be promoter, enhancer, or operator. And then here in the gene with the start codon right here, there would be exons and introns. And the exons will exit the nucleus and get spliced. And the introns would be non-coding DNA because they're not coding for a protein. What else would be non-coding DNA?
Like the poly-A tails.
Okay, so that would be what we would call, after, I should say, when the messenger RNA gets made, mRNA, there's what's called, here's 5' of the gene, here's 3' of the gene. There's called a 3' UTR, which is called untranslated region. And that would be maybe here, if here's the stop codon, here's the 3' UTR, and then the poly-A tail would get added after the 3' UTR. That's an interesting difference. Prokaryotes do not have poly-A tails. That's only a eukaryote thing. So that's another good, interesting difference that Wilson has brought up. What else is non-coding DNA?
We don't necessarily know that.
Yes, so if you have a chromosome, right, on that chromosome are genes, but there's all kinds of DNA that is like in between the genes, like here, and here, and here. We would call that non-coding DNA. And that's a big debate of what that does. A lot of people think that that's important for just structurally organizing the chromosomes, like a scaffold. Other people note that there's all kinds of studies that indicate that the non-coding DNA is important for regulation of genes. So like there's all kinds of looping effects where something will bind here and it will loop the DNA, and maybe you can only form that loop if there's a region of non-coding DNA. So there's all kinds of things in eukaryotic genomes. And again, it probably comes down to regulation because they're more complicated. More complicated things have to regulate their DNA in a much more precise manner. But prokaryotes, to say that they don't regulate precisely is incorrect also. Every organism has precise ways of regulating their genomic DNA. Okay, so I wanted to define, you'll see this word. I bring up words that you'll see in the literature. Streamlined. Okay, so what would a streamlined genome look like?
Like maximum efficiency.
Yes, or I should say minimal content. Minimal content, and minimal content translates into, not translates to protein, but translates into maximal efficiency. Maximal efficiency. So that would mean that if you look at a streamlined bacterial chromosome, okay, the regions between the genes is going to be very, very small, okay? And oftentimes the genes will literally be back to back. And there might even be only one regulatory element right here that regulates all these genes, okay? So these are streamlined, and that means the non-coding DNA is literally selected against. That means if you are a bacteria and you have extra non-coding DNA, that's a disadvantage, and you will be outcompeted by bacteria that have more streamlined genomes. Even more than that. So streamlined would be like its maximal efficiency, like Wilson said. What would reduced mean? A reduced genome.
Take out stuff that's not absolutely necessary.
Yes, so there are bacteria that have reduced genomes. That means like if you say the standard bacteria has about 5,000 ORFs, open reading frames, which encode 4 genes, okay? A reduced genome is gonna have like less than that, less than 5,000, okay? So that means they're actually losing genetic important coding content. They're losing coding content. They're losing genes. And oftentimes, can you think of any context in which a bacteria would want to lose a gene, or how it could survive after losing a gene? Right, because you're kind of trained to think bacteria, like all genes are useful, right? Otherwise they wouldn't be there. Like what would be the context in which you would want to lose a gene?
Like if they're in an environment where that gene's for like protection or something, and they don't really need that anymore. Yeah.
So imagine here's a really good example. That's a good line of logic. So here's a case of that. Imagine you are a bacteria in the soil, and imagine you are a bacteria that lives in a cell. Which one of these is going to have a more consistent environment? The cell, right? Because cells always, the cytoplasm is always actually like a fixed concentration. You're going to have always the exact same amount of ions. You're always going to have a certain amount of nutrients around in your environment. It's actually like a very consistent environment, provided that the organism you live in is alive, right? But as long as the organism is alive, it's going to be the same environment all the time. In the soil, you're going to be subject to all kinds of like weather conditions, whether or not the soil is rich or not, whether there's stuff growing around, other organisms, things like that. So things that are growing in the cell, like symbionts will often lose things. They'll lose genes, even essential genes. Maybe they'll even lose essential genes that are essential for synthesizing specific amino acids. They can no longer synthesize certain amino acids because all they need to do is steal them from the cytoplasm, right? They don't need to make that anymore. So they can actually literally lose those genes. So that's a case in which you would see reduced, quote unquote, reduced genomes. Okay, so I want you to kind of like get a range of knowledge of what's like a big genome, what's like a small genome. You should kind of have a working knowledge of actually kind of like quantities of genes. Okay, so a small virus, which would be maybe like a flavivirus, which would be like dengue or Zika, that's going to be like approximately like 10 genes. Okay, so you can make a machine that can replicate itself and do all the biological things except maybe like metabolism is iffy, but you can make a quote unquote organism or a machine with about 10 genes. Okay. There's a famous experiment. So Craig Venter is the guy who sequenced the human genome, and then he started his own institute in La Jolla, California, called the Venter Institute. And he has built a synthetic bacteria with the minimal amount of genes necessary to survive. Okay, so this is the bacteria with the single least amount of genes ever been created or observed, and that's about 300 genes. So in bacteria, at the very, very minimum, you need about 300 genes to survive and replicate that bacterial organism, and that is extremely reduced. Okay, small symbiont, like I was talking about, a small symbiont, which would be like Wolbachia. So I study Wolbachia, which is a symbiont bacteria that lives inside of insects. That's about 1,200 genes or ORFs. Okay, so it's a bacteria. It's definitely a reduced genome. 1,200 ORFs will allow you to get by and survive in that environment. And these are obligate intracellular, so they cannot survive outside in the dirt. They can only survive in the cell. So even with 1,200 cells or 1,200 genes, you literally can't survive extracellularly. You are dependent. These organisms are dependent on their host cell. Interesting, the JCVI minimal cell can grow by itself. It's not dependent on any other host cell. It can grow by itself. Okay, standard bacteria. So that's like E. coli, Serratia, things like that. That's going to be Pseudomonas, things like that, going to be about 5,000 genes. Okay. Let's see. So again, I'm just giving you the broad kind of understanding of like with a range. Okay, now you start to get to eukaryotes. So yeast. So yeast is like the quote unquote most simple, the model eukaryote, the simple euc. That's about 5,000 genes. So some of the most simple eukaryotes are about at the complexity of the more complex bacteria. Okay, and then you start to get greater than that, into greater than or equal to 5,000 genes. I don't know precisely. I think the human genome has like 21,000, something like that. I think it's in that order, but somebody should fact check me on that. And you can get like amoebas with like 120,000 genes. So there's a massive range of expansion in eukaryotes. Okay, but you want to kind of know that eukaryotes kind of start at about 5,000 and up, and bacteria are kind of like less than that. Euks. You kind of want to know that. That's the kind of complexity that you're going to be working with. I should also say, can you think of a case where like euks have many more genes, but they don't necessarily have like different genes. And what I mean by that is euks often have what are called paralogs. So like, for instance, hemoglobin. Hemoglobin is the protein in your blood that will carry oxygen throughout your system, right? Hemoglobin is a classic example of paralogs because humans have like 4 or 5 different versions of hemoglobin. There's like alpha, beta, delta. There's 4 different versions of it, okay? But they're all hemoglobin. They're all pretty much the same. So they're homologs, they're homologous, but because they're in the same genome, the same organism, they're called paralogs. Is that worth? We have enough time. I'll talk about that. So if you haven't, have you guys seen that before? Homolog, paralog, ortholog? You have. Do you know, could you define an ortholog?
No.
Okay, then it's worth me reinforcing the knowledge, right? Okay, so homolog means, in terms of proteins or in terms of genes, in protein that's going to mean the percent identity of amino acids. So like how many amino acids are exactly the same is pretty high. That means it's kind of like the same thing. You see the same structural primary organization. That's a homolog. In genes, it's going to be the percent identity of base pairs. So the exact nucleotide sequence is about the same. So you're kind of talking about the same thing. Okay, so like a good example is like the finger of a human is a homolog to the finger of a ape, right? That would be like a homolog, but that also translates at the protein level. Our genes actually are shared. So like we shared like 99% of genetic content with bananas, right? And that's because we have a bunch of homologs. And that's because basically the cell metabolism is pretty much the same from a human cell to a banana cell, even down to bacteria. So we share a lot of homologs even with bacteria. But in eukaryotes, you often see multiple copies. So those would be paralogs. So paralogs are homologs within one genome. So that means within your genome, you have 4 genes for hemoglobin. It might be more than 4, but I'm just giving an example. And those are all paralogs. They encode a similar thing, but the percent identity, they're not all exactly the same. They might only be like 95% amino acid identity. And that means they have some mutations, and that allows them to diversify, and that allows them to have different functions. So for instance, in the baby developing in a human, they actually express different hemoglobin because they need a more efficient way to get oxygen because they're kind of like taking oxygen from the mother's bloodstream. So that babies will actually express a different form of hemoglobin, which enhances their development in the womb. So like we have different paralogs. Paralogs allow specialization. And you will also see paralogs in bacteria. Like this is not a eukaryotic, prokaryotic thing. This is just a protein thing in general, okay? And then within homologs, underneath that, there's also orthologs. So an ortholog is if you have 2 species, same gene. So for instance, you look at monkey alpha hemoglobin, and you look at human alpha hemoglobin. These are orthologs of each other. Does that make sense? Okay. Now we can clarify that. Okay, structure of genomes. What do the genomes look like? Prokaryotes. So bacteria. Bacteria will, and this is not canon, okay, so you can find bacteria that do all kinds of different stuff. But in general, bacterial chromosome is going to be 1 circular chromosome. Okay? That's the general structure. And then they will also have what are called plasmids, plasmids. What are plasmids?
Extra little circles of DNA.
Yes, little circles of DNA. So maybe this one has 2 plasmids, plasmid A, plasmid B. Okay? And these encode little things. And these replicate just like mini chromosomes. Plasmids are basically like mini chromosomes. Okay, so the chromosome of reduced bacteria like Wolbachia is 1.2 mega, which is million base pairs. A plasmid is like, it depends, but the ones we use in molecular biology are usually about 5,000 base pairs. But you can get plasmids that go up to 20,000 base pairs. So that's kind of a range for a plasmid. It's kind of like, maybe I'll say like 3,000 to 20,000 base pairs. I'm trying to give you a good idea of the size of things, because when you architecturally build these things, size is very important. You need to kind of have a broader perspective of the size of things that you're working with, okay? Okay, so eukaryotes have bigger and more chromosomes. So when you think of eukaryotes, that's where you actually think of like, you look at the karyotypes and you see like chromosomes, right? So they'll have multiple chromosomes. So like mosquitoes will have 3, fruit flies have 3, humans have what, like 23 and 4 if you're, yes, something like that. Is that correct, 23? 23 pairs, right? Okay, so that's the pattern you'll see in eukaryotes. They have these chromosomes, okay? And they're separate. Okay, so that's kind of the basic structural organization of eukaryotes versus prokaryotic genetic content. Okay, so she just said, May just talked about diploid versus haploid. What's that? This is something you see in eukaryotes.
Diploids have 2 copies of every gene.
2 copies. And why? Because they have 2 copies of entire chromosomes. And haploids have 1 copy. What's aneuploid? Steven, what's aneuploid?
Do you know copies?
It could be no copies. It's basically kind of like, I don't know. Like, it's like it's not consistent is what aneuploid is. So like maybe you got 0 copies of chromosome 1, but maybe you got 10 copies of chromosome 5. It's like, it's all over the place. So cancerous cells will become aneuploid, and they will duplicate chromosomes that help them divide. And so you'll find cancerous cells, like HeLa cells, that have like 10 copies of the chromosomes. That's aneuploid, okay? And organisms can go through different phases, right? Like in our bodies, we have both haploid cells and diploid cells. What are our haploid cells? Yeah, the gametes. So a female will have an egg cell and a male will have a sperm cell. These become haploid through basic biology, meiosis, right? We all remember that. Diploid, most of our cells are somatic cells, are diploid. Do we know the difference between somatic and germline?
Somatic are like your body cells.
Yes. Body cells. Germline are what gets passed to the next generation, sperm. So their germlines are typically haploid, and our somatic cells are typically diploid. And things like yeast do, so this is all connected to sex. Yeast do sex, and so they make, they go through cycles of diploid, and then they sporulate, and become haploid. So things that have sex, you see this pattern where they have diploid, haploid phases. And right, the whole reason, am I at sex? Yes, I am. Okay, so sex. What are the benefits of sex? Why do organisms do sex?
Genetic diversity.
Yes, genetic diversity. And that literally, diversity. So that can give you an advantage of if you have different copies, you could maybe even call them paralogs. The more paralogs you have, maybe the more diversity you can kind of start generating. So you can generate genetic diversity, which can help you adapt. And this literally comes about from mixing, right? So when in basic biology, they teach you that your cells become haploid, and then when the sperm fertilizes the egg, the chromosomes literally will like, now you have a mixed organism that's half father, half mother, right? So the whole purpose of sex is mixing. You're mixing genetic diversity. Mixing is the great source of innovation. I think that's one of your knowledge, not facts. Or it should have been. Okay, let's see. DNA packaging. DNA packaging. So we've talked about size, okay? So we know prokaryotes have tiny genomes, and eukaryotes have big genomes. So how does that relate to DNA packaging?
If you're a eukaryote, you can have all your genes just out. You have to wrap them up.
Yeah, they literally do not fit. Very good. They literally don't fit. I can't remember what it is, but there's some like statistic, like if you stretch the human genome, it reaches like, I don't know if it's not around the world, I don't think, but it's something like phenomenal that will blow your mind. Like it's literally like will not fit in a cell. So eukaryotes have evolved elaborate ways of packaging, or you could call it folding, or you could call it winding their DNA, which prokaryotes kind of have, but they don't really like need it because their cells are so streamlined that they don't really need to package. So that's why in eukaryotes, you will find proteins like histones. Have you guys know what histones are?
They wrap around.
Yes. So histones, this is called the nucleosome. Histones are a complex. What's that mean?
It's a bunch of proteins.
It's a bunch of proteins. There's actually 4. There's It might be like a octamer of 4 of dimers. You know what I'm saying? So I think that's right. But there's I think there's like H2A, H2B, H3, and H4. So I think you're right. It might be an octamer. And in that case, they would dimerize. And then they form this particle, which is a nucleosome, which wraps DNA around it. I can't remember if it's 3.2 wraps. It's something like that. It's something on the order of that. And then there's this beads on a string model, right? And in between is a linker histone, H1, okay? Now beyond that, this gets wrapped into what's called the 30 nanometer fiber. I don't have these on here, but because I'm expecting you guys have kind of seen these before. You don't need to know, well, you do need to know them, but like I'm not gonna put this on a test. I'm just kind of going through it. Then there's 30 nanometer fiber, which are these beads on a string wrapped around. And then beyond that, to get to the chromosome structure, there are what's called SMC proteins, structural maintenance of chromosomes. Proteins. Clever name. So the SMC proteins, kind of like what they do is if you have all these 30 nanometer fibers, the SMC proteins will wrap around like this. They have a hinge right here, and then they have a clamp, and they kind of like hold these things together. So they are responsible for the structure of chromosomes, which is why they're called structural maintenance of chromosome proteins. So that's some more things. So in eukaryotes, you'll see histones organize the DNA with SMC proteins. And there's other things, but these are the major protein components of the chromosome. Okay, in bacteria, prokaryotes, they have their chromosome. And they have proteins that are similar. So they have a protein called HU, which if you go down back in the literature, it's histone, quote unquote, histone-like. I don't know how the U came in, but it's histone-like. And what it does is its function is it binds DNA and it wraps DNA and it bends DNA to basically what amounts to 180 degrees. So they have proteins that can bend and wrap DNA. And they are important for expression, they're important for the structural organization, but they don't fold and wrap their DNA like eukaryotes because they don't have to. They want it open. One of the patterns is, right, eukaryotes need complex regulation because they're so complicated. Prokaryotes want to be like moving and dividing quick. So they want their DNA out in open. So they don't want to close it up. Okay, so there's 2 kind of like fundamental differences in style of life that you'll see between eukaryotes and prokaryotes. Okay. So, how much time we got? 20 minutes. Oh my gosh, I put way too much information on me. Okay, so let's go through this. We know eukaryotes have a nucleus. Okay, eukaryotes have a nucleus. That means where does DNA get transcribed?
In the nucleus.
In the nucleus. Okay, so that means that the transcripts have to get out. Why do they have to get out? Where does protein get made?
Ribosome.
And the ribosomes, which are in the cytosol or the ER. So the mRNA has to get out of the nucleus. The exons have to exit. Okay, you don't have to do that in a prokaryote because there's no nucleus. Things in eukaryotes get spliced. We know what that is, right? Spliceosome, so you don't need to know the mechanism, you just need to know what it is. Like you need to know that there are enzymes that will take introns and exons and they will assemble them into basically an exon final 5 to 3' messenger RNA. And they do that through splicing. And you can also get different proteins by alternative splicing. So I'm kind of teaching you like the basic things you need to know. Like you need to know what splicing is kind of like what it is. And you need to know that eukaryotes have all these different systems that they can alternate the splicing. So they could make a completely different protein by splicing together different pieces, right? So that would be called alternate splicing. Okay. We do operons in the next lecture. I'm debating whether I skip this or not. I'm going to skip the operons for now. I might come back to it. Let me put operons for a question mark because we'll talk about that later more in detail. Bacterial promoters. Let's talk about promoters. And this is important for building stuff. If I get to the end and I have extra time, remind me about the operons. Okay, so you need to know about the promoters because the promoters are going to be turning on your gene X in whatever GMO you eventually make for your biotechnology application, right? So in bacteria, before we looked at that structure of a gene, there was a promoter, there was like a tatabox, and then there was a start codon ATG. Okay? In bacteria, the distance from here to here matters. If you move that start codon, one codon over, you put it here, and you change this to something else like TTT, your gene will not work. Okay? So the distance, and this is really important for the plasmids that you build. Okay? The distance, the precise distance from the promoter to the start codon determines whether your thing turns on or off. So oftentimes if you build a construct, you think it should be turning on gene X, it doesn't work, you sequence, and oftentimes you'll maybe find a mutation right around this region, okay? And it will break your gene. So what I want to say is in bacterial promoters, precise spacing matters, okay? Eukaryotic promoters, eukaryotes. So let's talk about yeast. If you build a yeast plasmid, it's not like that. You put a promoter here, it doesn't matter where you clone in your start codon. Let's say your start codon is here, let's say your start codon is here, let's say your start codon is here. As long as you're kind of like within like 1,000 base pairs, it's kind of gonna work. Okay, so in eukaryotes, spacing is kind of like less important, especially when you're building these plasmids that we work with. So you need to know that because when you start designing and constructing, if you build a bacterial expression plasmid, you need to program that precise start codon in a precise spot. If you are building something for expression in yeast, you can kind of put it anywhere. It doesn't really matter, okay? So you need to know that. That's a detail. Okay. So the models, just so that you know, the model eukaryote for biotechnology is going to be yeast. And the model eukaryote for prokaryotes is going to be E. coli. Okay? One of the reasons we use these things is because the yeast doubling time is 90 minutes. That means it can replicate in 90 minutes. In E. coli, it's 30 minutes. So they are fast. That means if you do an experiment with E. coli, it's ready to go the next day. If you do an experiment with yeast, it's ready to go in 3 days. That's fast. If you do an experiment with Drosophila, it's ready to go in 2 to 4 weeks. If you do an experiment with mice, it's ready to go in 6 months, maybe a year. Okay? So we work with these things because it's fast, and you can precisely dissect hypotheses very, very quickly with these model systems. That's why we work with them. Speed is king. That's one of my knowledge, not facts. Okay. Gram-negative versus gram-positive. Do you guys know that? Are you familiar with that? One or two, but not all. Okay. So gram-negative. This relates to the surface of prokaryotes. Okay? The surface of a prokaryote is on a sliding scale from gram-negative to gram-positive. And it has to do with the thickness of their cell wall. So if you look at a gram-negative bacteria, you can find bacteria like mycoplasma. So the JCVI minimal 300 cell is mycoplasma, and it has no cell wall. So it's literally just a thing with no cell wall. You can find bacteria in the middle, which are considered gram-negative, like Wolbachia or E. coli, that have a surface structure that's in between. And what they have is they have an inner membrane. They have a layer, which would be called a thin cell wall of peptidoglycan. This is a chemical that cross-links. So it forms a hard structure, a wall, literally a wall. And then they have what's called an outer membrane. Double membranes with a cell wall. That's classical, kind of like intermediate. Okay? So mycoplasma is considered, so here's where it gets tricky. And because it's a sliding scale, and people pretend that it's like this dichotomy, like there's gram-negative and there's gram-positive. No, it's like a sliding scale. And there's some bacteria that are gram-negative that have no cell wall, and there's some bacteria that are gram-negative that have a tiny cell wall. And then gram-positive is when you have an inner membrane and a thick cell wall. Thick cell wall. Okay? So it's like a sliding scale. So what you want to know is you just kind of want to know that keep in mind that the structures of the surface of a bacteria differ. Dylan, does a gram-positive have a outer membrane?
I think so.
I have to check this. Did somebody Google this? Who's got a computer? Google does gram-positive have double membrane? Because I'm blanking on that right now. And I need to be fact-checked.
So for the gram-negative that are in the middle, they have an inner membrane,
An
a
outer
layer
membrane.
of peptidoglycan, and then the outer membrane.
Yep.
Okay.
No, they don't.
Yeah, that's what I thought. So gram-positive, the other definition is there's no outer. They just have a massive, thick cell wall and an inner membrane. So the great key to the gram-negative is their double membrane, but tiny cell wall. And the gram-positives are thick cell wall with one membrane. Does that make sense? And again, it's actually kind of difficult. I've been studying bacteria for 10 years, and it still confuses me because people set it up as this dichotomy, which it's not. It's a sliding scale. Okay, let's move on. Why do we build stuff with bacteria? It's fast. They're like little farms. So if we want to study protein X, we have to actually physically have protein X to do experiments on. Does that make sense? So we need a way to actually like make it. So we utilize bacteria and we coerce them by giving them unlimited food to basically produce protein X for us so that we can study it. That's why we use bacteria. But there's also more reasons. I think we talk about them in the plasmid section. But that's in general kind of what you want to think. They're little tiny farms that grow things that we want them to grow if we teach them right. And teaching them is all in the programming of your plasmids. Okay, so let's get into the logical questions of basic biology, the underlying structures of why things are happening. Why are bacteria simple? Why?
They don't have mitochondria, so energies.
You read the paper. Okay, good. Let's get to that in a second. Let me first just start off by saying, so bacteria are living life in perpetual starvation. When you grow bacteria in the lab, you're breaking that rule. You're giving them a soup to feed off of. But in the wild, bacteria are perpetually starved. That means they're perpetually under selection to survive. That means they will not waste energy. That's why they are selected for the maximal efficiency, the streamlined or even reduced genomes. Okay. Now there's a little caveat: is if it's going to depend on the niche. Okay. So for instance, why would you have bacteria that have 6,000 to 7,000 genes or 5,000 genes, and then you have some bacteria that are 12,000 genes? It's going to depend on the niche. So like soil bacteria, they might all of a sudden be in the soil and they're starving, and then all of a sudden a flood happens and it gives them a whole bunch of nutrients, and they need to capitalize on that immediately. So then they change the entire like transcriptional processing to different subsets of metabolism. So they can kind of like adapt, okay? But the more simple ones, the very, very simple ones, are less versatile. So I will say there's kind of like this dichotomy between versatility, I should say trade-offs, not dichotomy. There's a trade-off between versatility and efficiency, right? You want to get rid of things you're not using, but if you get rid of something you might use 5 years from now, then that means you might die 5 years from now. So that's kind of like the trade-off between versatility versus efficiency. And this is a constant battle in evolution that people actually like study. Okay. Now let me give you a scenario. Let's give you a scenario of a population of bacteria. Okay, you have green bacteria with a genome of 500, let's say 5,000 genes, and you have yellow bacteria with a genome of 1,000 genes. Let's make a case, and let's say these bacteria are equal in ability to use the nutrients that I give them. So I put them in a soup, wash the soup over them, and I come back a day later. Which one is going to have more babies?
Yellow.
Yes. Why?
They have less DNA to replicate before they split.
Yeah, they're literally going to be faster at replicating because they have less DNA. So if you want to be fast and you want to replicate fast, you want as little DNA to replicate as possible, because I told you and I taught you that those enzymes that regulate the polymerases that regulate and add on the base pairs actually have a speed limitation. And they can only add so many base pairs per second. The Q5, the fastest one, is the, what was it? It's 1,000 for like 15 seconds, something like that. And no, it's faster. I think it's 1,000 every 5. It's fast, something like that. But there is a speed limitation, okay? And if you have a tiny genome, you can replicate faster. And then this bacteria wins after 1,000 years because there's none of this left. It's all been outcompeted. Does that make sense? So there is this constant pressure. The answer is that speed is king. E. coli replicates so fast. Oh, this is super interesting. So E. coli actually replicate so fast that they divide their genome before the genome has finished replicating. So what they do is they have a chromosome and they start replicating their chromosome for their granddaughter. So they will replicate, start replication one time for their daughter. And then right after that, they start replicating a second time for their granddaughter. Okay? So they're replicating multiple chromosomes in one cell so that once it divides, it's actually got a second chromosome for the daughter ready to load. That's how fast, that's how much they care about speed. They replicate for their granddaughters. I already did that. So speed is king. Okay, the other thing is, why are they so simple? Okay, and this gets to the reading if you read it. So let's do a euc. This is good, we're almost finished. And a proc. Proc is going to be tiny, and a euc is going to be big. That's the other thing is size. Eukaryotes are big. Okay? You need to know that the cell surface, literally the surface area, is the limiting factor for absorbing nutrients. All nutrients that feed the cell have to come through the membrane. So the surface area is a limiting factor of how much nutrients you can absorb and how fast you can absorb those nutrients. And I'll say, a bigger cell or a tinier cell, which one do you think is going to use more energy?
Bigger cell.
The bigger cell has higher metabolism. It's going to be making more stuff. It costs more energy. This one is going to be less. So there's a key measurement, the surface to area, surface to volume, surface area to volume ratio, okay? Ratios. And that's important. So this one there's literally like a size limitation. You can't grow to an infinite size cell because at some point you hit a limit at which you literally cannot absorb enough nutrients to support your cell growth. So now the question is, so prokaryotes are literally limited to a tinier size. They cannot grow beyond a certain size because they hit that surface area to volume ratio, their maximal limit. But eukaryotes can grow to higher size. How? How have they solved the problem?
Mitochondria.
They actually have mitochondria inside them, which are literally tiny bacteria. I mean, I'm simplifying, but they're tiny bacteria that make energy for them. So they have solved the problem of they don't have to absorb nutrients from their surface because they have internalized tiny bacteria that make energy for them. So that's why eukaryotes have bigger cells and can form multicellular organisms because they have these mitochondria. And if you read the reading here, I'm running out of time, but if you read the reading, the Power, Sex, Suicide book, which is an awesome book, you should read it. The concept, the basic theme of that book is power, in terms of the mitochondria, allows you to have the energy to do sex, and actually at the end, the power that the mitochondria create in the cell kills you in the end by damaging your DNA. That's kind of like the thesis of the Power, Sex, Suicide book. But it has a really good chapter on Eukaryotes versus Prokaryotes, which I wanted you guys to read. Let's see. What should I focus on for the last 2 minutes? So, how did the first eukaryote evolve? So there were bacteria. How did the first eukaryote form? Well, the first eukaryote was something bigger that swallowed something smaller. Okay? And mitochondria have their own genomes. If you actually look at the mitochondrial genome and you blast that, you compare that to other genomes of bacteria that we've sequenced, it's actually closely related to a clade of bacteria called Anaplasma tassiae, which includes Wolbachia, the one that I study. These are alpha proteobacteria. So there's actually really good evidence that the eukaryote first came about by being a cell that ate another cell, and this little cell inside learned how to survive as an intracellular parasite, but then eventually streamlined its genome to a point where they were kind of like symbiotic. And Power Sex Suicide book has a good chapter on that as well. I don't think I attached that one, but there's a good chapter on that. Okay, so let's end there. Great work.